Proteins are a class of biologically important compounds. They are condensation polymers of about 20 different alpha amino acids, which are linked by peptide bonds. A peptide bond is formed when the amino group of one amino acid molecule interacts with the carboxyl group of another amino acid molecule. This reaction involves the elimination of a water molecule. Thus, the interaction between the amino group and the carboxyl group of two of the same or of different amino acids results in the formation of an amide linkage, that is, an NHC double bond O group called a peptide linkage. The product formed when two amino acids interact is called a dipeptide. The name dipeptide indicates a peptide bond that is formed between two amino acid molecules. For example, when glycine combines with alanine, a dipeptide, glycylalanine, is formed. Similarly, when two glycine molecules combine, glycylglycine is formed. Note that, by convention, the amino end is shown on the left and the carboxyl group is on the right of the resulting dipeptide. Also note that the peptide bond is planar. The CON and H atoms are all in the same plane along with the alpha carbons. Let's work on a practice problem. Show the formation of the peptide bond that forms between alanine and serine. Keep the amino acids in the order given here. It may help to start with the peptide backbone. Then look up the side chains and include them in the correct order. The alanine-serine, represented as alicer dipeptide, results. Three amino acids linked by two peptide linkages are called tripeptides. The tripeptide glutathione protects the cells from oxidizing agents. It is formed from glutamic acid, cysteine, and glycine. Similarly, when 4, 5, or 6 amino acids are linked, the products obtained are called tetrapeptide, pentapeptide, and hexapeptide respectively. When more than 10 amino acids are linked, the product obtained is called a polypeptide. Thus, depending upon the number of amino acid residues present per molecule, peptides are classified into dipeptide, tripeptide, tetrapeptide, and so on up to polypeptides. Polypeptide chains that have either more than 100 amino acid residues with a molecular mass greater than 10,000 or less than 100 amino acid residues with a well-defined conformation are referred to as proteins. Let us now look at the classification of proteins. On the basis of molecular shape, proteins are broadly classified into two classes: fibrous proteins and globular proteins. In fibrous proteins, the polypeptide chains are long and thread-like and run parallel to each other to form fibers. These parallel chains are held together by hydrogen bonds and disulfide linkages. Due to their strong intermolecular forces of attraction, Fibrous proteins are generally insoluble in water. Important examples of fibrous proteins include keratin, found in hair, nails, skin, etc. Myosin, found in muscles. Collagen, found in tendons. And fibroin, found in silk. Thus, these proteins form the structural material of animal tissues. In globular proteins, the polypeptide chains coil around to give a roughly spherical shape. These are soluble in water due to the presence of hydrophilic groups. Globular proteins maintain and regulate life processes. For example, insulin regulates the blood sugar levels in the body. Hemoglobin and albumin are some other examples. Let us now discuss the three-dimensional structure of proteins. The structure of proteins is studied at 4 different levels, in the ascending order of their complexity, as primary, secondary, tertiary, and the quaternary structure. Let us first discuss the primary structure of proteins. The specific sequence of amino acids in the polypeptide chain reflects the primary structure of proteins. Here, we see the specific arrangement of amino acids of the enzyme lysozyme. Every protein has its own unique amino acid sequence. Any change in the sequence of amino acids creates an altogether different protein. The secondary structure of proteins refers to the arrangement of the polypeptide chains in space. One important type of secondary structure is the alpha helix, illustrated here in a ribbon diagram. Each polypeptide chain is coiled to form a spiral structure called alpha helix. The alpha helix is a right-handed helical chain in which the hydrogen bonding occurs between different pairs of the chain to hold the helix together. The hydrogen bonds occur between the oxygen atom of the carbonyl group of one amino acid residue and the hydrogen atom of the NH group of a different amino acid residue on the adjacent turn of the helix, as shown here. Another common secondary structure in proteins is referred to as beta-pleated sheet structure. The beta-pleated sheet is nowadays referred to as the beta structure. The arrows in the ribbon structure here show the beta structure. The contraction of the peptide chains results in a pleated sheet. The chains lie side by side and are held together by hydrogen bonds. Such a structure is called a beta structure or arrangement. The tertiary structure of proteins describes the further folding of the secondary structure. The tertiary arrangement of helices and sheets is held together by hydrogen bonding, disulfide linkages, van der Waals forces, and electrostatic forces of attraction. Shown here is the tertiary structure of proteins. The further folding of the secondary structure gives rise to 2 major molecular shapes, fibrous and globular proteins. Let us now look at the quaternary structure. Some proteins consist of multiple polypeptide chains called subunits. The quaternary structure describes how the subunits are arranged in space relative to one another in the complete protein. Hemoglobin is an important example. As shown in the illustration, the 4 protein chains of hemoglobin are arranged in a compact bundle. A protein found in a biological system with a unique structure and biological activity is referred to as a native protein. In some respects, proteins are rather fragile. Temperature or pH changes can cause a disruption of the hydrogen bonds. When hydrogen bonds break, the globules unfold, the helices unravel, and the beta sheets flatten out. This is called